And Human Genome Project means what you can say during this project. What did scientists? They have sequenced all genes which is present into the human genome. They sequenced means they find out the sequences like ATGC, ATGC. Total sequence find out which is present into the human genome. Okay, so this is called the Human Genome Project. Okay, now it is started with the one agency which is known as the National Human Genome Research. Okay, National Human Genome Research Institute and NIH, Nutrition Institute of Health. Okay, so two agencies together involves. One is the National Human Genome Research Institute, and second is the NIH. So these two together involves the Human Genome Project. When it was started in 1990. Just look at here. This is the only story, like not a very important thing. And what is the important I will discuss, that is the how many genes present in the chromosome, the smallest chromosome, largest chromosome, how many genes are found into our genome. Okay, so these things they have given. Look at here. Okay, US government started Human Genome Project in 1990 by the Coordinated Department of Energy and the National Institute of Health. So I told already, National Institute of Health and DEA, Department of Energy. Okay, so Department of Energy, DE, and National Institute of Health. So together they involve. Now genome. What is the genome? Genome, the whole hereditary information of an organism that is encoded by the DNA called a genome. Sometimes like that, or sometimes they have also given haploid set of gene. Haploid set of gene. This is also called the genome. Anyway, whatever they have given, you have to learn it. Haploid set of gene, or the whole hereditary information of an organism that is encoded, was called the mega project, because it have the too much cost. It have the too much cost, and it was very big project, so that's why it is called the mega project. Human genome has approximately 3 into 10 to power 9 bases. Cost of sequencing required 3 US dollar per billion. Okay, per base pair, 3 US dollar. Clear? And how many base pair is there? 3 into 10 to power 9 base pair. Means too much base pair, and each base pair cost is the 3 US dollar. 3 US dollar each base pair cost. So that's why 3 US dollar means, are you getting? That means you can say 3 US dollar means 8 3s are 24, means 300, approx 300 you can say. So 300 per pair cost, per base pair cost came. Okay, so this have given. Again, the total estimated cost of the project is 9 billion US dollar. Okay, so the cost was very high. How many? 9 billion US dollar, because already they have given the 3 into 10 to power 9 base pair. Clear? How many base pair is there? 3 into 10 to power 9 base pair. So in this way, the total cost of the project is 9 billion US dollar. Okay, the total cost of the project is 9 billion US dollar. Why? Because they have already given 3 into 10 to power 9 base pair, and we know each base pair they have the 3 US dollar cost. Okay, the obtained sequence were to be stored in the typed form in books. Okay, they have stored in the form of, means they have the typed, and in form of books they have stored. Okay, and if each page of the book contained 1000 letters, that means they have stored in the form of book, but each page of the book they have how many letters? 1000 letters. Okay, and each book very big. Okay, this is the mega project. Why? Because they have the 1000 letters, 1000 pages, and 3300 books. Okay, 3,300 books needed. So to store this information, that's why it is called the mega project. Okay, aims of our goal of the project. I told already, firstly to sequence the gene and their function. If you have sequenced the gene means you can find their function. Okay, so here they have to identify the approximate 20 to 25,000 genes in the human genome. How many total gene found into the human genome? That is the 30,000 gene. 30. Just wait a minute. How many gene found? 30,000. 30,000 genes found into the human genome. Previously, they thought the human gene may be 85,000 or 1 lakh. But after the sequencing, he got how many gene was there. So only 30,000 gene was there. Okay. So this again to identify the approximate 2,025,000 genes in the human DNA. Okay. Okay. Now, to determine the sequence of the 3 billion bases that make up the human DNA. Okay. This was the object, means why they are doing, firstly to identify the how many genes. They are thinking 20 to 25,000 genes in the human DNA. Approximate, but previously they understood 85 to 85, means 80 to 85,000. Next, to store this information in the databases that we have seen that book. Okay, in the book they have stored. To improve the tools for data analysis, very important. What he did to improve the tools for data analysis. Data analysis means for the human beings we can find out which type of genes inherited metabolic disorder, or SLR, sex-linked recessive disorder. So all this activity we can find out with the help of this thing. Okay, to address the very important, to address the ethical, legal, and social issue. This is the very important ELSI. This is called the ELSI, ethical, legal, social issues. Okay, ethical, legal, social issues. That is called the ELSI. Okay, so to address the ethical, legal, and social issues that arise from the genome research. Okay, because that time they are doing the research and they are going to count the human genome. Okay, so maybe ethical, legal, and social issues will come. So they have the to address this. Okay, means they have informed we will not do which is not good for human health. Okay, because the science is good. Science is good if you are doing. Science is bad also if you are going to misuse. Clear? I told science is good if you are doing, that is good, but it is also not good for human society. Okay, suppose that some person they culture the embryo. You know one important story that Chinese people. So Chinese peoples they are eating the embryo. Embryo means the child which is found inside the mother. Okay, fetus. Okay, so what they observed that, okay, they observed that means ethical, legal, social issues. They can't do those work which is not good for human population. Okay, that is the ELSI, ethical, legal, social issues. Okay, so here they have given the average gene consists of 3,000 bases, but sizes varies. Okay, average gene means each gene have how many base pair? Each gene have the 3,000 bases. Okay, suppose that one gene, how many base pair? 3,000 bases. Okay, 3,000 bases is present. Okay. But size can be vary. Bases number will be the same. Size can be vary. Means the length protein. It is a type of protein synthesized by the DNA. because we know that transcription translation. So during this, firstly DNA will transcribe, after that translate, and ultimately it will form the dystrophin. Okay, now the total number of genes estimated 3000. It was lower than previous estimated 80 to 1,40,000 genes. Okay, I told firstly scientists what he understood. He understood that total estimated gene is the 80,000 to 1,40,000. They thought the total gene is the 80,000 to 1,40,000. Okay, but after HGP, what he got the result? He got the result, the total number of genes found into the human genome is the 30,000. Okay, the total number of genes found into the human genome, that is the 30,000 gene. Okay, 30,000 gene found, but previously estimated, what he estimated? He estimated that the total number of gene was the 80,000 to 1,40,000. Okay, now almost 99.9% nucleotide bases are exactly the same in all people. Okay, in all peoples the bases are 99% is the exactly same. There is no variation, because you know the DNA 98.5%. They have given the 99. You have to know the 98.9. I will also here they have not mentioned in your NCERT. That is the molecular marker. Molecular marker which is used for the identify the particular location of the gene, that is called the molecular marker. So molecular marker we can also use for this. Okay, but here in your NCERT they have not given, so that's why I will not discuss about the molecular marker. Okay, so almost 99.9% nucleotide bases are exactly the same in all people. Okay, it is the same whether human beings is they have the same 99.9%. Okay, little bit variation. The function are unknown for the over 50% of discovered gene. Okay, they have discovered 50% and 50% function, now they have unknown. They also don't know the exact function of the 50% genes. Okay, because the function are unknown for over 50% of the discovered gene. Okay, they have discovered total 30,000 discovered. Okay, in 30,000, 15,000 they know what is the function of this gene. Okay, but remaining 15,000 they don't know what is the function of this gene. Okay. Less than 2% of the genome codes for the protein. I told 1.5%, 98.5% they are the similar. 98.5% it is the similar. 98.5% bases are the similar. Junk DNA only 1.5%. They have given less than 2% means 1.5%. Less than 2% means 1.5%. So 1.5% of the genome, 1.5% of the genome. No. Okay. So less than 2% of the genome codes for protein. That I told how many percent? 1.5%. So 1.5% of the total genome which codes for the protein. 1.5% codes for protein. And 98.5% it is known as the junk DNA. Now, this is the different base pair, and this is the chromosome number 1. How many base pair is there? 246 million base pair. So that is too much. Okay. So here they have given. That is the smallest. Y. It is the smallest. I think 1 is the largest and it is the smallest. Means Y is the smallest chromosome. Short stature homeobox, Y link short stature. Larry dysteriasis is different gene responsible for the different function. Okay. Now another one, repeated sequences. Repeated means that repeated sequences make up very large portion of the human genome. Repeated means similar sequences. So 98.5, I told this is the similar or repeated sequence which form the large proportion of the genome. Chromosome 1 has the highest gene. I told already highest number of the genes. Chromosome 1 has the most of the genes. That is most 2968 genes. And Y has the fewest gene. This you have to learn it. Okay, this is the important. Already PYQS. This I have already asked in your NEET exam. So how many number of the genes found in chromosome first and how many number of genes found into the chromosome last, that is the Y. Which have the highest number of genes is found into the chromosome 1, 2968, and while fewest number means lowest number of genes found into the Y. Scientists have identified 1.4 million locations where single basis DNA difference, and this is called the SNPS. That is called the SNPS, SNPs or SNPS. Some places they have given the SNPs and some places they have given the SNPS. That you can say single nucleotide, single nucleotide, single nucleotide. Wait, wait one. Single nucleotide polymorphism. So this SNP, it is the universal and maximum distributed molecular mark. SNP is the maximally distributed molecular marker. Okay, SNPs. They have given the scientists have identified about 1.4 million locations where the single basis DNA differences. This is called the SNPS. They have given SNPS, single nucleotide polymorphism, or it is also known as the SNPs. Okay, so SNPS or SNPs. This information helps finding chromosomal location for disease associated sequence and tracing human history. Okay, this means this DNA sequences or base pairs, they help to tracing the human history. That means you can say phylogeny. We can trace the phylogeny of human beings with the help of these genes. Okay, means this human genome project we can find the tracing the human history. Next is the DNA fingerprinting. Okay, now next topic, that is the DNA fingerprinting. It is also very important. And DNA fingerprinting, we have to see. I will tell one small story that you can learn how the concept was there. What happened? One person, he has the buffalo. Okay, he has the buffalo. Okay, this is the story about Haryana state. This is the story about the Haryana state. So he has the buffalo. What happened? Next day, this buffalo she was producing high quantity of the milk, means 100 liter per day. 100 liter she was producing 100 liter per day milk. Okay, so thief some thief came and they stolen the buffalo. Okay, they stolen the buffalo. Next day, what happened? Buffalo was not there. He went and he claimed into the court. Firstly, they have tried to search, but he didn't get. So ultimately, he went in court and claimed. Okay. So firstly, tried to search, but few days later, he didn't. So firstly, he tried to search, but he didn't get. Ultimately, what happened? 1 year or 1.5 year later, what he got? He saw the buffalo. He saw the buffalo, and he said, "This is my buffalo." He saw the buffalo, I think 200 or 300 kilometers away from their native place, and he saw the buffalo, and he said, "This is my buffalo." But thief said, "No, it is not your buffalo. It is mine buffalo." Then that owner who have this buffalo, he said, "I have the child. I have the child of that. I have the child of that buffalo. So I can go for the DNA fingerprinting." Okay. So he claimed for DNA fingerprinting because child already there. That buffalo child already owner have, only buffalo mother lost. Okay, so ultimately it went into the court, and court they performed the DNA fingerprinting. They performed the DNA because why they are going to perform DNA fingerprinting? This buffalo was very costly. That buffalo price is 6 to 7 lakhs. This was very costly, so that's why they did the DNA fingerprinting, and ultimately he got the same buffalo because she was the mother of that child. So through the DNA fingerprinting, it is clarified that the buffalo is the means mother of that child. In this way, he got your buffalo. Okay, he got your buffalo, and he claimed means, and that person who was the culprit, they also uh, they have also did the act through the IPs. Okay, okay, leave it. So this is the story of DNA fingerprinting. Now let's start about the DNA fingerprinting. Okay, DNA fingerprinting technology. This was firstly evolved by the Alex Jeffrey, who have discovered in India. You know, do you know who have discovered the DNA fingerprinting technology in India? Do you know anybody who is considered as the father of Indian DNA fingerprinting? Anybody? You don't know? That is Dr. Lalji Singh. He is from my native home. He is from my native hometown. Dr. Lalji Singh. Okay. Dr. Lalji Singh. Lalji Singh. Lalji Singh. Okay, so Dr. Lalji Singh, who is considered as a father of Indian DNA fingerprinting. Okay, father of Indian DNA fingerprinting, that is Lalji Singh considered. Okay, so here Alex Jeffrey, who is considered as a father of DNA fingerprinting, but father of Indian DNA fingerprinting, that is the Lalji Singh, which works into the CCMB, Center for Cell and Molecular Biology, Hyderabad. Okay, this is the research agency which is located into the Hyderabad Center for Cellular and Molecular Biology. Okay, so I will talk this into the next class. That is the last topic of this chapter. After that, this chapter will be over. Okay, so DNA fingerprinting, then chapter will be the over. Clear? Okay, so thank you to all of you.
Thank you, sir.
Okay.
Thank you.